series called MENDEL 2.0. It's organized this autumn semester by Theory of Interactive Media, Faculty of Art, Hauser University. I would like to invite here Dr. Jens Hauser from University of Copenhagen, Department of Arts and Culture Studies and Medical Museum, Faculty of Health Sciences. That will have a whole lecture on staging aliveness is biomedia, from living statues to genetic fingerprints and designers' Pigeons. Short CV to introduce our guest now. Dr. Jens Hauser is a Paris and Copenhagen-based art curator and writer. As a media studies scholar, he is focusing on the intersections between art and technology. He holds a dual research position at both the Department of Arts and Cultural Studies at the Medical Museum, Faculty of Health Sciences at the University of Copenhagen, and is distinguished affiliated faculty member of the Department of Art, Art History and Design and Michigan at Michigan State University. His curated exhibitions include La Bibliothèque, Nantes 2003, Still Living, Perth 2007, SK Interfaces, Liverpool 2008, Luxembourg 2009, the Article Biennale, Stavanger 2008, France Biotics, Riga 2010, Fingerprints, Berlin 2011, Munich 2012, Synthetic, Vienna 2011, and Assembled Standard Minimal, Berlin 2015. Hauser is also founding collaborator of the European Culture Channel Art and has produced numerous reportages and radio features. The lecture is part of the invited lecture series called MENDEL 2.0, organized by Theory of Interactive Media, Faculty of Arts, Hauser University, by which we celebrate 150 years anniversary since Mendel's lecture, which he presented to the society, the Nature Society, Science Society in Brno in 1865. Mendel's perspective, observation, talent, and use of mathematics stood behind the benefiting of three principles often referred to as Mendel's principles of heredity. And we would like to show by this series of invited lectures that it's a kind of basic principles that stay in the beginning of scientific fields like biology or genetics, but also it's very much inspiring for new media art, different projects and works of art. So I would like to invite here Dr. Jens Hauser.
The title I have chosen has several reasons, of course. The first reason of this title is that I'm going to talk today about the artists who enter the labs today, that they are those fascinated by recent scientific advances as such, and also including them as artistic media, but also that they relate to the long history of art in which the staging of aliveness has always been a central occupation. So in times where biotechnological art gets interested in synthetic biology, and we have the democratization of lab tools that leads to the appropriation by tinkerers, tactical media activists who apply the critical potential of open source culture from the digital age of media art to grassroots DIY biology and biohacking. We have a new kind of trend for not only hard and software but also to wetware art. And in the sense, I mean, it's probably not prominent in every country, but right now in Denmark, for example, we have had this program together with Synergy Biofaction in Vienna, that we set up the first artist residency in a synthetic biology lab for artists wanting to work with protocells. And we have chosen the Turkish artist Orkan Telhan to work with edible artistic protocells. They are all, just recently we have picked for this residency that we set up in Los Angeles at the UCI, University of California, Irvine, another artist in residency program for which we have been choosing first Anna Dimitriou as an artist who is going to be part of this lecture series, as I understand, also chosen Dimitri Gelfand and Evelina Domnich, who are from Russia and Belarusia. Also, we can say that, well, there's still a very limited amount of exhibitions, and it's still very difficult to see if this works. It's very complex to set up. It's technically challenging, it's expensive. The staging techniques are not really museum friendly. So, but still, there are increasingly exhibitions that we managed to organize worldwide. And just this year, I have personally curated 2 exhibitions. The first was SO3 in France, that doesn't refer to sulfur trioxide as a pollutant, but to 3 significant others, usually despised, neglected biological better halves, so to say, concretely viruses, bacteria, and plasmids. And all the artworks were about virus, bacteria, and plasmids, but real virus, not representation or simulation of. And so, as today science is focusing on the benefits of bacteriophages, microbiome studies, or decontaminating bacteria, we have artists turning these techniques upside down. The other exhibition was Assembled Standard Minimal by Revital Cohn and Tur van Baalen in Berlin, and they discussed the notion of artificially created sterility in this area of mechanized and standardized live manipulation techniques. And for sterile, that we can see here, it's in fact goldfish that they had made by a Japanese biologist that are engineered to actually be born without any reproductive organs, because they are manipulated to do so-called surrogate productions. And on the other hand, they constructed this machine which is automating the process of producing sterile fish, so that in fact it's a kind of outsourcing of reproduction to the machine. Very absurd kind of coupling of artworks. But on which history is this curatorial, this cultural, this artistic staging of aliveness grounded? And I propose that we first start with art history in this sense, because many of the artists are not only interested in the freak show kind of event, or they are not interested necessarily in pushing science further or only subverting contemporary science, but very often there's a kind of drive to try to have new forms of how to stage aliveness, which is a central feature, of course, in art history. So, starting, for example, with a revealing story from Giorgio Vasari, tells from the end of the 15th century on a commission that was made to Leonardo da Vinci, who, in order to produce a perfect painting of aliveness, collected snakes, lizards, and bats so to translate their body parts into dragon-like chimera paintings, and forgetting, by the way, about the rotting animal carcasses in his studio, so that when the client arrives and comes to pick up the painting, Leonardo's lifelike painting, plus the smell, makes the client flee in panic, because it's not only visual, but it was also stinking. And this anecdote was told by Vasari, and it's interesting because it is indeed this enlivenment, this artistic animation, that was then considered as a prime quality in early modern art, both by Leonardo and by his biographer Vasari. But what was at stake at this time was the appearance of life, the possibility of a fictional enlivenment, not the actual animation. So Leonardo da Vinci is quoted, "Painting is not alive, but expresses living things. So where natural vitality is missing, one must create an artificial one." So I want to follow here art historian Frank Fehlenbach's argument that Leonardo's example, within the context of early modern art, in fact represents an oscillation between lifelessness and life, and this points to 3 different directions which are today of great importance, especially given that Leonardo, combining organic studies and machine building, is also often quoted as a key figure of an integrated art and science practice. So, in the concept of enlivenment as an aesthetic appearance, not only of actually bringing artifacts to life. This is contrasting with earlier ambitions known from the moving automata, or cult images, iconic images which were there in order to be actual presence, to stage presence and not to be representation. But Leonardo derived this concept of the oscillation between lifelessness and intended and life from biology itself, because Da Vinci considered that in fact the organism itself is constantly dying and at the same time revived again through the pulsation of the blood. So the organism then is always living and dead at the same time, growing and decaying, and therefore living art also should ambivalently embody this characteristics of an organism itself. But third, toward the end of his life, Leonardo turns his back on painting. He's stopping painting and gets involved in real assemblage of hybrid organisms from different real organic body parts. I'm saying this because I want to focus on the notion of enlivenment or staging of aliveness, meaning different strategies succeeded each other to stage vitality, either by form, material, or process. And it has also been historically debated all the time which adequate techniques and media to present or represent, to simulate or animate aliveness should be employed according to the then dominant epistemological context of the time. And my argument is that even when we look at art forms today, we have this split into representation, concept-based traditional contemporary art, could be painting or sculpture. Then we have a presence of phenomenon-based dry media art, let's say with robotics or informatics. And likewise, we have now today in a third step presence and phenomenon-based wet media art that employs biotechnology, including synthetic biology, and leading to what we can call a kind of contemporary paragoning, so they will call the competition between different art forms. So, since the earliest anthropomorphic statues, myth of vivification surround artifacts made by the artist's hand. As we know, the animation of malleable matter stands in a long picture tradition, Pygmalion here. And from this 19th century, the biological metaphor is also continued in the discussion of the artwork itself as an organism. That's a big discourse in the 19th century or the beginning of the 20th century. So by means of form, material, process, a touch of aliveness is staged, aiming at involving the viewer viscerally. So art has imagined, represented and mimicked, then simulated and quite recently manipulated living beings and systems for real. So after painting, sculpture, automata, etc., art of the late 20th century has first employed dry informatics and robotics to stage aliveness, and as well since shortly wet cell and molecular biology. Transgenics, the synthesis of DNA sequences, so-called biobricks, molecular biological visual imaging techniques such as gel electrophoresis, DNA chips and so on, cell and tissue engineering, the reuse of retroviruses and the cloning of bacterial plasmid DNA belong to a repertoire of a still marginal but very experimental form of contemporary art today. So it has become, to have a kind of conjoint definition of myself and Eduardo Cach, a process-based art of transformation in vivo or in vitro that manipulates biological materials at discrete levels, so individual cells, proteins, genes, nucleotides, and creates displays that allow audiences to partake of them cognitively and also emotionally. So this left image is from the first bio-art and hands-on workshop for artists that I attended by Symbiotica, organized by the Arts Catalyst in London. And this is a strange formal analogy, isn't there? No? You have a, it's very different. I mean, despite the formal analogy, to just read an image, you have here the black habits in Rembrandt's painting, you have the white lab coats, so obviously the expertise system and the belief systems have shifted also from the black habits to the white lab coats. And also, this is just a snapshot, and the image itself is not supposed to be art, it's just an amateur photography, while on the right hand, the painting itself is in fact the artwork. And here, the artists, in fact, themselves are just portrayed as those who are in fact gaining expertise to actually learn how to manipulate living systems at a micro scale, not at the body scale for real, so miniaturization and so on. And the changing of the status of the images, of course. So in my contribution today, I want to focus on 4 key aspects. First, that I pretend that we have art today in what I have coined an epistemological turn. So to say that we ask more how to, not only what we know, but how we know what we know. And I'm reluctant to think of these practices simply as art and science overlappings, aiming at overcoming the supposedly split between what P.C. Snow once coined the 2 cultures. I didn't want to argue that these practices partake of this epistemological turn when art itself is less concerned with formal aesthetic principles but increasingly linked to technical and scientific critique, alternative knowledge production, focusing not only on what we know but how we know what we know. So artists being themselves professionals in constructing and deconstructing of metaphors and both constructors of both senses and sensations. Second aspect that I'm going to illustrate, I hope, is that we have an oscillation between the technologization and the animation of the technological. So that is concerned with what are actually the appropriate media to employ when we stage a license. The technologization of the animate would mean the instrumentalization of organic systems or beings, and on the opposite side, the animation of the technological. That means the staging of lifelike processes or entities in other than biological media. And this oscillation seems to be very close to another aspect of top-down versus bottom-up technologies. We'll come to this back later. The other point is that we can think of biomedia today as a continuation of traditional media functions, and I will illustrate this in the last part of the section. And also then today, what does it mean that we have DIY and biohacking? And I will relate the latter as a strategy of tinkering as opposite to purposeful engineering. So all these reflections were central to me and my scientific partner Markus Schmidt when we curated the exhibition Synthetic at the Natural History Museum in Vienna in 2011. It presented a large scope of artworks related to the new field of synthetic biology, beyond the fashionable use of just biobricks. And we wanted to focus also on the ethical consequences of artists dealing with synthetic biology, and also to focus on the historical and epistemological roots. Just an example here, these are protocells that Rachel Armstrong, a British architect, artist and medical, is staging in an installation called Living Chemistry. And it shows the live formation of protocells that are precursors or very primitive living cells that are just formed by chemicals and which share some of the physical chemical properties of living cells. But interesting also that it's accompanied by an animation film here, and this natural history of protocells. Here the subtitles suggest a kind of dialogue between the cells. They are emotional narratives, and they of course illustrate also the human desire to even see in the smallest units of emerging life, to see this through an anthropocentric lens. Likewise, that we had on stage, very invisible by the way, in small liquid, maybe 1 or 2 millimeters, so it's also the question of how do we actually stage that if it's so small. Is it interesting if you stage that? So here, James Tours and Stephanie Chanteau's anthropomorphic molecules are named nanopulsions. So they are in fact performing dancing cooking and so on, but they are just anthropomorphic molecules, and you could in 2 milliliters of liquid have billions of these molecules without being able to actually see them. On the other hand, you have a kind of top-down approach with creating artistically biobricks. That is an example here by Tour van Dalen's Pigeons project, in which he is attempting at making pigeons shit soap by modifying the metabolism of the bacteria that occur in the gut of the pigeons with the help of a specifically designed biobrick that is a standardized genetic building block collected in the Registry of Standard Biological Parts set up at the Massachusetts Institute of Technology. And one of this biobrick is lowering the pH level in Bacillus subtilis, and other makes it express lipase, that makes grease digesting, fat digesting enzymes. So to add new functionalities to animals that are commonly seen as flying rats, so turning them potentially into swarming urban disinfection machines. Van Dalen has also designed two functional objects. The first one is a pigeonry to be attached to the windowsill, potentially allowing for the pigeons to be fed and enhanced, and the second, a kind of interface for a parked automobile, allowing pigeons to land and shit soap onto the windscreen. So what is interesting is that at the same time there is a practical, technical investigation of what can be done, but also a metaphorical one and a deconstruction of the metaphor, because it's about the design of absorbed artifacts that echoes metaphorically the design of genetic circuits in this very dominant engineering talk that is so present in synthetic biology, where it's talked about as circuits of genetic circuits, of chassis, not of living organisms, not of living beings, but of modules, of standardized parts, a vocabulary much inspired by a concept called orthogonality, which is coming from computer science and meaning that unlike in most living systems, you can produce technical elements where one component doesn't create side effects on the other element. Just like changing the winker in a car doesn't affect the steering, right? So Van Dalen here is ironically deconstructing the metaphors, the language metaphors, also by proposing this absurd artifacts. Now, in art, especially in media art, the notion and the fascination with so-called artificial life has been very crucial over the last 30 years. And it's worth to have a look at the strong opposition between the proponents of first the animation of the technological and those of the technologization of the animate. And we can take them as epistemic indicators. We can observe that there is an antagonistic, not very friendly relationship between the animation of the technological, proposed by what I call the carbophobics, and the technologization of the animated by what I call the carbophiles, or sometimes even the carbofetishists. We all know this book here that was very influential for media artists, especially in the 90s, by Christopher Langton, 1980s, when it was concerned with artificial life, simulation and robots, following Christopher Langton's much quoted manifesto from 1980, saying that artificial life is a study of man-made systems that exhibits behaviors, characteristics of natural living systems. It complements the traditional biological science concerned with the analysis of living organisms by attempting to synthesize lifelike behaviors, but within computers and other artificial media. And further, since it's possible to abstract the logical form of a machine from its physical hardware, it's nature to ask whether it is possible to abstract the logical form of an organism from its biochemical wetware. So artificial life should therefore be, quote, extending the empirical foundation upon which biology is based beyond the carbon chain life that has evolved on Earth, therefore locating life as we know it within the larger picture of life as it could be. Ironically speaking, and nobody has noticed this before, and just reviewed in my PhD, by the way. Ironically speaking, Langton seems to be so ironic to carbon that he even misspells the word carbon on the first page of his manifesto and amputates the word carbon of its R, as if he wanted to annihilate the organization function of the carbon atom as such. But the text has been so much a foundation for a battle between the carbophilic and the carbophobic in relation to art, that we may have a look at what artists now do with this manifesto when they define what artificial life or bioart should be in their mind. So, first opposition here, let's see what carbophobic artist Leonardo Maura, himself constructing painting robots, affirms. He says that bioart is a new kind of biologically inspired art that campaigns for the emergence of new artificial, dynamic and self-sustainable nature. The main point is to generate life as an artistic expression, but not life as it is, but life as it could be, we see the quote. And this new kind of art departs radically from the sad idea, you can hear nearly crying here, the sad idea of using human and animal bodies transformed in artworks, as well as the practice of employing organic materials in the pieces and installations that have plagued 20th century museums and art galleries. So you can very much know where he stands. As an opposite, carbophile artist Eduard Katz, who has introduced the term transgenic art in 1998, claims that bioart is a new direction of contemporary art that manipulates the processes of life, and bioart employs several approaches that could be the coaching of biomaterials into a specific inert shapes or behaviors. Second, the unusual or subversive use of biotech tools and processes. And third, the invention or transformation of living systems from a single cell to another. Is it in this organic sense that bioart uses the properties of life and its materials, changes organisms within their own species, or invents life with new characteristics? So we can understand that the visual animosity between both tendencies of the carbophobics and the carbophiles, the first accusing the latter of anachronistic materialism or the lack of complexity, and the later blaming the first for naive digicentrism, inadequate art media, and the blind belief in programmable genetic code-based processes beyond the particular materials. And when we have a look at the practices that on both sides are emerging, they are of course very different. Right? And all this has to do, of course, also with how we define life. And we know that's not a technical definition, but a philosophical definition. And the definition very often that in German biophilosophy is taken is the one that is satisfying very many criteria by Bernhard Rensch, who has been listed these in his book Epistemology-Based Biophilosophy. So we have to know, and we have to actually look at this, what kind of characteristics of living beings are emphasized, when, how and why. And even when artists do this, are they more focusing on individuation, temporality, regulation, metabolism, behavior, and so on. So all this can be a lexical or indices for living systems, but it's often enough to take a few, to mix them up, and to come up with different medias. Now, this is not necessarily new either, and I have argued together also, and that's why I take the example of this brilliant text that Jessica Riskin has written on similar cases of antagonistic dialectics in the 18th century. Jessica Riskin has demonstrated how the illusion of liveness is created though through, on the one hand, the behavior or movement, and on the other hand, through material organic aspects on the other. So in her text, 18th century webwork, very intriguing type by the way, Riskin pays attention to the counter-model of animating machines, which generated the illusion of life, not only by simulating activity, but also by, quote, soft and moist substances, rubber, leather, cork, fluids and airs, pneumatic systems combined with organic-looking exteriors and simulated material metabolisms. So she's opposing in this text also, for example, Maillard's mechanical swan from 1733, and nearly at the same period, Vaucanson's famous duck from 1738. But while Maillard's mechanical animal just paddled through the water and its exterior aspects not really reminded an actual swan, it was just to represent but not to simulate a natural swan. By contrast, Vaucanson's duck was staged with organic metabolic activity. It not only flapped its wings, but seemingly digested grain and rejected excrement, which is indeed a process that was later demonstrated to be fraudulent. But still, for the viewer, it was important. The duck was eating and the duck was shitting. Very important. So now, transpose this into the 20th century. We encountered a similar twist in the 20th century, and here, in the light of cybernetics and robotics, computers were first used to simulate emergent and intelligent behaviors, such as in Gray Walter's turtles, wheeled turtle-like mobile robotic vehicles equipped with sensors and able to exhibit complex behaviors and to explore their environment, or as a kind of artwork that many of you know, of course, Ken Rinaldo's Autopoiesis here, a whole genre of media art installations that emerged, robotic sculptures that interact with the public, modify the behaviors based on feedback from infrared sensor and dependent on the presence of the participant. Oh, this one here was also at Ars Electronica some years ago, this fabulous artwork, this robotic chair, which is featuring self-repair and self-assembly that are also characteristics of artificial aliveness. So here we have algorithms which govern the chair's behavior and enabling it to learn from its environment. So it's collapsing and building it up from scratch anew. But it's of course not in the very organic term of what aliveness here is about. Then there's an opposite. Let's take this one. Functional organic metabolic processes are at the center of Vindebois' shipping, digesting machines named Cloaca, and in this series of bio-cybernetic automata, the artist programs the grammar of the complex fecal language of enzymes and gut bacteria populating the artificial digestive tract. A performative installation, and visitors often applaud when the actual droppings are falling, right? So it's really performative. And the Levois effect renders transparent and makes central that part that was in fact fraud and hidden in Vaucanson's canard mécanique. So why I wanted to bring these precursors here into play is that in synthetic biology today, software, hardware, and wetware seem to meet in an unprecedented fashion. And synthetic biology is currently also being approached as a discipline in which the top-down and the bottom-up, the virtual and the actual, and where simulation is being conveyed into synthesis. Not only simulating, but synthesizing. So when it comes to DNA synthesis, for example, to DNA-based biological research on minimal organisms to test biological units reduced to their minimal functions just necessary to their survival, minimal genome, protocells, xenobiology with its functional biological systems that are not yet existent in nature but are not even intended to interact with natural systems. Interestingly and strikingly, the term itself is already 100 years old and has been coined by French natural scientist Stéphane Leduc, La biologie synthétique. As in physics, he said, observation, then analysis, should be followed by synthesis, even in living systems. So they have a kind of medial development, an epistemological development, where he was predicting that biological would fulfill the same paths. First, observation, then analysis, then synthesis. And as Manuel Landa has outlined in his book Philosophy and Simulation, the emergence of synthetic reasons, the increasing capacity of simulation has itself become the very motor for synthesis. We simulate more in order to synthesize more. So for artificial life today and artificial life art, this means that simulation and rematerialization should not be divided, but wetware compatible to be media adequate. And artists recently have been increasingly combining the technologization of the animate and the animation of the technological in a very fruitful way. And some people who may have been to the Ars Electronica Festival this year have remarked that Gilberto Esparza from Mexico actually won the Golden Leica, the most important art prize in media arts probably, in our category, which is hybrid art, precisely with not this piece, but which a new piece, but which is a follow-up piece from this Plantas Nomadas, so nomadic plants. And here this piece already was conceived as an autonomous robotic ecosystem, but also that is containing bacteria in microbial fuel cells, and thanks to the energy production, can make plants that are growing on the robot make walk, to make them motile. So it combines hardware, software and wetware to purify polluted water, filter out chemicals, heavy metals, release oxygen, generate energy and become self-sufficient in the process. So today living machines are no longer imitations or simulations in non-organic materialities. And here, when Gilberto Esparza's Plantas Nomadas are said to express autonomous and intelligent behavior, this might not be meant to describe the mimicking of human cognition, but rather the system's decentralized intelligence to clean up humankind's mess in times of major ecological crisis and atmospheric crisis. Interestingly, this is maybe also a kind of epistemological indicator. You probably all know this very important competition that was on for many years of the VIDA, Art and Artificial Life International Competition. And it was probably the competition with the most generous pot of money for artists, not only for the prize money, but also to produce artists' works. And what is right now happening is that this award has been stopped, and there's probably reasons for that. So Esparza's work, to me, is indicative of a more profound shift towards wetware, and this can be observed in this decision of the Spanish Telefónica company to stop its Art and Artificial Life competition, which Esparza, by the way, has won some years ago with this robot. Precisely, the award has been initiated to highlight projects associated with robotics, electronic avatars, chaotic algorithms, nobots, cellular automata, neural networks, computer viruses, virtual ecosystems, hybrid systems, telepresence, augmented reality. But there was a clear increase of projects that were awarded that were challenging the very boundaries between the living and the non-living, including carbon-based life, genetic engineering, and tissue culture, self-experimentation in biology and so on. So Telefónica may not really wanted to be associated with this shift towards wetware anymore. So we can fully assume that wetware-based practices are gaining importance as artistic media, and we might want to see how far these instances of biomediality, as they call them, fulfill functions of previous media, electronic, physical media, and how they are used to stage aliveness. So in my own concept of biomediality, I have outlined that media based on physical principles can be continued, they can be thought through the shift towards bio and convergent technologies, conserving existing media functions and adding new capacities, self-repair, adaptation, evolve, but biologically. So I split them into three categories, and I then will give you three examples of how this can actually unfold. The first one would be just to think of media in this very traditional sense of where milieu coming from. So milieu as an enabling condition. Yeah? If you talk to a scientist in the biological lab, when you say, give me the media, he is not going to switch his television on, but he is giving you the pot with a nutrient medium. This is a medium for a biologist. And in this very sense, the milieu is a very old definition of what it is to have an enabling condition. The second one would be the very typical definition of media that we have, media in the sense of means. Media in the sense of means of transformation or generation, so to have the ability to transmit, store and process, for example, and to shift this from the electronics, from the physical to the biological realm, making use of living systems, internal mechanisms to transmit, to store, to process. That can be genetically modified organisms, it can be bioinformatics, can be DNA or cell computing prototypes. This would fall in the second category. In the third category, this is to be media in the sense of instances of measurement. So we always had optical measurement devices in the line of traditional media of perception analysis, such as optical or physical instruments. But here, in which one biological entity is measuring another one. So to say what is happening in DNA chips or what is happening with gel electrophoresis. One organic system is measuring another one. It's not physics, it's not chemistry. So these biomedia can actually overlap. And this is based on the assumption that media in general can be conceived as what has been called a loose coupling of atomistically split and arranged elements that produce forms. But in biology, with cells on organic macromolecules as their crucial smallest units, even de- and reorganized elements themselves still remain structurally relevant. That's why the carbon atom is so important, because this is something that has itself as the smallest unit already in organizing function. So I want to just illustrate very shortly the three categories of what media or biomedia can be by three short artistic examples. The first example, called Victimless Leather, by the Tissue Culture and Art Project, employs biomedia in the very sense of the milieu. It's an enabling condition in tissue culture. So here the artists grow stitchless, miniature-like leather jackets that are grown out of immortalized animal and human cells that form a living layer of tissue on a polymer matrix. And Victimless Leather is staging the ideal of producing leather without having to kill an animal. But here the leather is not a dead surface, but a living trans-species interface between the cells. And these prototypes are being grown live in incubators and techno-scientific glassware bodies. But we have to ask, is the promise of victimless culturing not really contrasted by the visual aspect? It seems to have kind of Christic image, that's a kind of victimized, suspended body that is there. But even more important is that it gains the effect from the quality of the media not appearing as media. Because here the media is actually the message. The media is a message in the very McLuhan sense of the term, because to get this neutral medium, this is not victimless. You have to suck it out of this fetal calf serum, which you need to do tissue culture. You suck it out of a living heart of a living cow. So actually it produces victim, but the abstraction is very, very high. So this is what people usually do not see. So you only can read the work actually when you know how this medium is actually produced. So it's not victimless. So the victimless is the title. So it's a trompe l'oeil in a sense, and you cannot read the work. That's why it has epistemic qualities, because the supposed victimlessness is contradicted by the very biomedia themselves. So you need to be part also of this network of epistemic knowledge. The second, and of course this is interesting, because if you leave it for a while, they get polluted, they get contaminated, they have fungi, they have bacteria, and so on. So as a curator, of course, this is a kind of challenging and very expensive exercise. because you are spending half of your, when you do it not as video, but if you do it with the real pieces and you fly the artist in and out, you spend your money on airplane fees and of continuously regrowing, rotting ephemerals. For the shown sculptor faces, in Luxembourg, for example, the artist had to come 3 times from Australia to set up another collection of cell garments. The second example, concerning the second category, media as means of transformation or generation to implement new functions. Here we have a piece by the Japanese artist Jun Takita, Light Only Light, which is a transgenic artwork which takes the form of a light-emitting moss sculpture in the shape of the artist's own brain. So he has done a magnetic resonance scan image of his own brain that is then 3-dimensionally printed in plaster, and the surface being grown over with transgenic bioluminescent moss, developed employing a technique similar to biomarkers that are used in science. So he is in fact using this genetic sequence from a firefly that has been transferred into the Fiscum mitrilla patens moss. And the firefly enzyme luciferase is then activated with luciferin solution to produce oxyluciferin and energy in the form of light. So presenting us with a plant that emits light, that is visible to the naked eye in total darkness, such in a still life painting, the suite with this artwork presents the transgenic as a very ambiguous cognitive achievement of the human brain. It is allowing for the creation of plants with the ability to emit light in a way that normally only animal species can do. And natural evolution has not produced organisms both able to consume light as energy and to emit light. So the human observer here, who himself relies on oxygen that are produced by plants, can contemplate the glowing in darkness, whereas mosses are photosynthetic, so they require themselves light to conduct photosynthesis. So they do the total opposite of what they actually want to do. But then it's also, when it comes back to artistry, for me it's a kind of vanitas motif, right? Or a kind of memento mori, or we can think of it also as pointing to historical representations such as Albrecht Dürer's Saint Jérôme, in which the reading and thinking Christian apologist is simultaneously pointing his finger to his own head and to the skull on his opus text. So it's a total memento mori quote in a certain sense. Also, the viewer has to come extremely close to the artwork. It's a kind of immediate percept that is depending on the viewer's own co-creative posture and make him aware of its own limitations. And finally, also, there are other links to art history we could talk about for hours, but for Takita, this is a kind of miniature garden architecture. And he makes the role of the body of the perception of the participant so central because he sees the sculpture as a miniature landscape in which the brains, the convolutions, equate the ornamental patterns in 17th century French garden architecture, such as built by André Le Nôtre. And André Le Nôtre, as we know, also very often employed a system which is called perspective ralentie, which is a slowed down perspective. And this slowed down perspective was a strategy employed always by Le Nôtre in gardens. You can see this also in Versailles, that the dimensions of the garden patterns are tailored to precisely match the eye and the receptive position of the visitor in order to counteract this optical distortion. So things that are far away seem to create the illusion that elements are actually moving closer. So here Takita transposes this desire of illusionism and to create more nearness and to ask what is actually an experience where everything what we perceive can be manipulated. But manipulated in terms of perception, but also in terms of genetics. The last artistic case study I want to mention is the third category I mentioned, that of the classes of biomedia that represent instances of measuring other things, where one biological entity is measuring another. And I want to give this example of artist Paul Vanus, who challenges the very notion of the so-called genetic fingerprint. And you know maybe the techniques. This is meaning employing agarose, gel and electric current, DNA samples, which are always molecules that are negatively charged, are drawn toward the positive voltage end of the gel, and depending on their molecular size, smaller molecules travel faster than larger ones, and therefore you have this abstract banding patterns that occur. But now, DNA is here employed in Paul Vanus's work not to represent DNA or to have images of DNA, but make actually images out of DNA, physically, materially. So, from the mindset of a political artist, Vanus subverts the indexicality that is supposed to be inherent in the very notion of the DNA fingerprint. But it's not an imprint. It's not an imprint, but it's a trace. So why do we talk about even the genetic fingerprint? Isn't this expression itself is misleading? And precisely, it's not an imprint, it's a trace of the body, which doesn't need to come from the finger, and split hairs, it's not even genetic, as it's coming also only from the introns. So Paul Vanus says that, this genetic fingerprints are understood to be single human identifiers, but also it's a cultural artifact. We make this up with our techniques, and we do not disclose really the protocol. So Paul Vanus is asking why such metaphors as genetic fingerprinting are hiding away their technical construction of the supposedly neutral. So are these genes ontologized in order to be used as an argument for nature predetermination, leading scientists to search even for genes that are responsible for homosexuality, criminality, in the same way that photography was used once to draw conclusions about characters, about traits on the basis of physiognomy. So what photography was for physiognomy, Paul Vanus says today, there may be kind of political biased science as well that we have to question critically. So on this latent figure protocol, analytic laboratory methods, so that we have in the third category, they measure something, they're used for synthesis. So instead of making images from unknown DNA samples, he's using images he wants to be predictable from known DNA samples. So he's generating iconic images which are symbolically highly charged, ID 01, cross and skull and crossbones, chicken and the egg or whatever. So he is treating them like in a row of pixels and wanting to downgrade the scientific authority of this objective or supposedly objective DNA fingerprint. Similarly, in an exhibition I curated with Paul in Berlin some years ago, here in this Suspect Inversion Center, also called SICK, Suspect Inversion Center, sounds a bit like CSI, right? Crime Scene Investigation, you know? Or SICK. SICK in scientific literature is the Latin sic errata scriptum, which means this thus it was written, so which normally indicates that we have a problem in the quote. You know, it's a wrong quote. So here, he is in fact quoting other people's DNA. So in this Subject Immersion Center, he is creating a functional laboratory in which he is creating genetic fingerprints from criminal cases such as O.J. Simpson's DNA. So he's in fact creating O.J. Simpson's DNA only by using his own DNA. So it is, so to say, kind of molecular photoshopping. And we had these accusations in the 90s when Time magazine proposed this poster image of O.J. Simpson and was accused to digitally having darkened O.J. Simpson to make him look more criminal, right? So the darker he was, the more criminal he looked, and therefore this was photoshopping, and now we have molecular photoshopping as art techniques. So So just examples. But also Corvo News is currently also into the biohacking culture. All this gel electrophoresis, or also the PCR, this molecular photocopying, he can do it on a kind of campfire. And that's what he did here, deep woods PCR. How can I create these techniques actually with very primitive techniques? So here he is inspired also by the current biohacking culture and making 3 different temperatures of water on a campfire, which brings me to the last aspect of my contribution, notably the subversive use or the hacking of biotechnologies. This is not that new, in fact. It's supposed to be, according to literature of people who have not been around before, occurring around 2007, but you already in 2004 and earlier you have these kind of push for subversive use of biotechnology for hobbyist kids and personal biocomputing since more than 10 years already. Here we have just a manual by Natalie Jeremienko and Eugene Becker about biotech hobbyist culture. It's only in the last 5, maybe 5 years that artists, creators, hackers and tinkerers have been appropriating so-called biobricks to implement new functions into model organisms such as E. coli bacteria, to aestheticize them, and these building blocks are always prone to humorous or subversive design products such as the E-Chroma project by Ginsberg and King, where engineered bacteria, secret colored pigments used as bioindicators. It is striking that the now popular event such as the IGEN competitions, the International Genetically Engineered Machine competition, advertises synthetic biology as open source concept, borrowed from computer and internet culture. So they aim at emphasizing this collaborative culture, shared program, as the opposite side of soft and wetware owned by great corporations. But it needs to be asked, of course, whether this cool dressing up of synthetic biology is not also a clever way to de-dramatize a technology that critical observers have called extreme genetic engineering. And in the case of the IGEN competition, the ambiguity between these promoted technologies, the community practices stemming from digital culture, seem to be voluntarily entertained. So, but doesn't this biobrick-focused approach lead us also to a genetics or code-based centered view? And here it has to be stressed that citizen science DIY approaches, or biohacking, very often more emphasize an open hardware movement, not that much an open software movement, but which is happening side by side. When you look at the Oxford Dictionary, it says that hacking is a piece of computer code that performs some function, an alternative or addition to a commercial program. So biohacking, of course, evokes subculture, anti-institutionalism, and now also the combination of soft, hard, and wetware. And it needs to be questioned whether this analogy with the movement of the computer hacking since the 60s is appropriate, or if it first and almost represents an attempt to integrate emerging bio-practices within the tradition of communication-based digital media art. And it also needs to be analyzed what lurks behind the opposition between the happy hacker, the ethical hacker in this DIY community versus the evil and narrow-thinking, purposeful engineer. So most biohackers I know reject these code-based approaches, such as in the official science, here in this paper, for example, by Antoine Danchin and Victor De Lorenzo, have asked whether synthetic biology actually is a new discipline. If it's just doing and performing new kind of genetic engineering, then it's not really new. So they say, well, is it a new word or is it just new words? And they call for a specific European perspective, more integrative, that is combining different fields, engineering, computing, modeling, but also molecular biology, genetics, traditional biotechnologies, origins of life, and also protocell research, protein modeling, and so on. But more generally, where does a positive image of tinkering come from, as opposed to purposeful engineering? I have maybe one kind of hint to that. Here, this very famous artist from article from 1977, François Jacob himself, who is often quoted as the inventor of the program metaphor in molecular biology, has argued that the evolutionary process of natural selection should not be described by the metaphor of engineering, but by that of the metaphor of tinkering. He's writing that nature, as a molecular tinkerer, quote, would slowly modify his work, unceasingly touching it, cutting here, lengthening here, seizing the opportunities to adapt it progressively to a new use. Unlike engineers, he says, tinkerers who tackle the same problem are likely to end up with different solutions. The tinkerer gives the materials unexpected functions to produce a new object. From an old bicycle, he will make a roulette, and from a broken chair, a cabinet of a radio. And with this in mind, we have a different look of an art piece that was first. Herzog Einberger, maybe one of the science historians, very much pushing for this. It's connected to acknowledges non-human agency a lot. So I do not see them as opposite, but rather coming up together, personally. Does it answer what you...
Yeah, that's basically like for me this is like a new field which I'm not really like oriented in, so I was just like curious about like how you reflect on
Well, really a lot of artworks are about the mingling, no? For example, I'm right now curating in Denmark a festival about what I have described as micro-performativity. No? The different notions of what performativity was, from linguistics to feminist to technological critique, and all these notions of anthropology, theater studies, what performativity has gone through. And micro-performativity being acknowledging, in fact, what you call the ontological term. No? That the actors, let's say, are plasmids. That the very agency is lying not in the hands or in the brain of human cognition. We have created recently a big bacteria research group at our university, which is internationally spreading now, so that we claim that after big data we have big bacteria, and this is the term what we use, that it's a material term, right? And we're learning from bacteria, and the next exhibition for which our museum has got and won the Danish Museum Award in competition with 48 other museums, it's investigating precisely the effect of gut bacteria on the brain. There will be a whole exhibition, art and science exhibition about this, with the title Mind the Gut. Not Mind the Gut, but Mind the Gut. So I think it's part of both this epistemological thinking, but also thinking of non-human agencies, which is, I think, very related to what you wanted to mention.
Yes, may I have a question about Davis radio? Because I still don't catch the meaning or the the relationship of that mechanical part and the biological art. If there is a violent disturbance of this biological, which disturbs the mechanic part of, or it's a kind of cooperation, or is what kind of relationship is there?
You mean technically speaking, or in the more metaphorical sense?
Metaphorically, yes.
I mean, Joe is very interested in hobbyist culture, no?
Yeah.
Radio amateurs, and I remember when driving with Joe around in Boston, in his very antique car, right, that he got for $1 or $20, and he only fills up this fuel when somebody pays him five gallons of fuel. That we were listening to radio there, and I told him, well, a lot of your artworks is not really easy to understand because we don't go to art museums with sequencers, right? And it's not very fancy and not very flashy and it's not very sexy to have a petri dish in the fridge, right? While all the concepts in there. He said, well, this may change. Hey, listen to this radio, you know? It's the last station I can capture with my radio because all are digital and I don't have a digital receiver. So this is the last station I actually capture. So the radio metaphor was there to say, in 10 years, everybody will have sequences at home. And this will be the technique. While with this old radio, I cannot continue listening to music. So it's about acceleration and about a medial shift towards web media or biomediality. This is one aspect. The second is to promote hobbyist culture, tinkering culture, because this is what companies who want to sell amplifiers and machines, they want to sell the machines. They don't want you to look inside the machines. So he's militant in the sense of training people to look inside and make something different with it. So if you have a kind of radio circuit of this kind of platine, this very primitive crystal radio, which gets its energy even not from our electricity source, but potentially for the ambient waves, right? So to use very complex genetic engineering to produce a completely obsolete piece of technique of electronics. This is a reversing of the metaphor. While you would think that synthetic biology is making the circuits in a very sophisticated way in biology. It didn't work though. It's nice work and he got awarded for this, but I have never listened to it. I mean, it didn't work.
For me, it was very interesting that Leduc, Stefan Leduc, it was in 18th century? 19th century. He told that first we have to observe, analyze, and then synthesize. It's very important for this biomedia art. And also it's kind of metaphor for all this age.
Yeah, he was in the times of chemistry and physics. He said, well, why should it only happen in physics and chemistry? So he was doing crystal gardens. This was also the reason why to the exhibition in Vienna we invited Roman Kirchner. And Roman Kirchner was this very nice piece that I don't have an image from like Roots, but you know it. Actually, it could be, yeah, it's growing, it's crystals, it's a computer that is actually not only modifying its own software, but also its own hardware. And this is based on the chemical computer that was imagined by Gordon Pask. And Gordon Pask thinking of these kind of chemical computers that was self-assembly, and not to have this hard and software split.
I would like to ask you, because most of the new media artists, scholars or curators, they have background in different fields of studies. Like they come from the traditional art, history, education, or some other fields. And I would be interested what is your background, because it seems to me, I was very impressed with the taxonomy of the live art, and it seems to me that your background is in the visual art. Am I right or not?
My actual training is in media studies.
Media studies?
Yes. But
like traditional media studies?
Well, what is traditional? I mean,
it's
not communication, not necessarily communication, but in the German sense, media studies, it's very much influenced not only by, let's say, Flusser, but also by, of course, guys like Zielinski or Kittler. So it's a material studies that is very close to epistemology. Right? So in the kind of, let's say, Anglo-Saxon approach to media studies, very often social complexity and political issues, cultural issues are central. And in this, let's say, since the 80s, in the German media studies, the angle of actually opening the computer. You know? Kittler once said at Transmediale, well, all this shitty media art is HTML, pseudo-computing art, I really don't like that. I mean, who has not opened his computer, you know, I don't trust him.
Okay. And how do you get interested in this kind of art?
I think I was when I was younger, I wanted to study biology, but actually studied theater, cinema, communication, media studies instead of biology, and then psychology as well, sociology, and then in France I studied science journalism. And then I was part of a team that created this German-French European cultural television channel Arte. So I was one of the team that was in the producing the pilots in '92. And so I was a journalist for 15 years. And then I was following all those media art festivals. And I was going there and I saw all this digital art, all the Ars Electronicas, Transmediales, all the festivals. And then in the middle of the 90s, I saw that, whoa, it's getting wet. You know? It's not only about simulation anymore, it's not about screen-based, which was interesting at this time, but then it came back that, well, there was this interest for real biology in mind. So it was a way of getting the kind of reconciliation, reconciling with what I didn't study. And then you can catch up with, I think, technical knowledge a little bit. So yeah, I studied art history as well, of course, and film history and philosophy. But then you bring it together. And what I like, for example, in Zielinski's approach of deep media and deep time, or also what's happening with media archaeology, you know? Also what Jussi Parikka is writing is very relevant. I think it's why we are greening the media, what is the anthropomorphic in the very neglecting of the material qualities of communication techniques of digital media. So there is this material turn in there. And I think it's not random that even all these digital thinkers less think now only about social communication effects of humankind, but also get into this more maybe ontological and new materialism. And, yeah, and I think it comes together sometime. Whatever this rematerialization.
Just another question, if I can. You mentioned the term molecular photoshopping. For me, it sounds very malicious, and it has much to do with biopolitics, biopower, and all this DIY biohacking. It sounds friendly, but according to me, it could have a violent results, or
technology is always a double-edged sword. I mean,
yes, but are there some restrictions or some jury that controls, or this kind of activity?
you mean the artist or the biohacking scene?
Biohacking. This DIY, someone opens a
Well, there are manifestos. You know, you have a
small lab and create some bacteria.
Well, you have ethical codes. You know, you have ethical codes.
Yeah.
And I think that every DIY space has an ethical code. You know? That's the first thing that you get when you go to such a space. You know? To say, these are the rules of conduct, and we don't do evil, and we have an ethics, and so on. And I do not think, or I do not know of anything evil happening there. But you probably also know that the FBI was, in fact, the FBI was the institution worldwide who created the first worldwide biohackers gathering. You know this? No. Because they were afraid that in these labs everybody's going in very freaking directions. So the biohackers didn't have money to make a big gathering, but the FBI invited them all to the US and come together. Please tell us, and if you see something, say something, and trying to get information, you know. And so a lot of biohackers actually accepted the invitation from the FBI because it was the first possibility to know the other guys. Right? And I know several of these.
And there was no disguise. It was like officials said, okay, we are inviting you and so on. Right?
Well, I mean, what you. It was a way, probably as a strategy to say, okay, we don't tell you everything, and but it was say, well, don't imagine that it's so freaky what we do, right? So it was not to be harassed like Steve Kurz was. You know, all of this critical art ensemble case when Steve was actually risking 20 years of jail because he had harmless bacteria at home, right? So in the Americas, at least, artists know how far this pressure can go if you're really under the radar. No? And there was this big campaign when it happened to Steve that it was costing thousands of dollars every month to fight these accusations of bioterrorism and then of mail and wire fraud. No? So it's easy to put the charge on you. So I think many of the hackers also played the game to say, well, you disclose a little bit, stay friendly, leave us alone, nothing happens, you know. But it's actually funny that they put a lot of money to invite all these biohackers from abroad to fly them to, was it San Francisco or something? Yeah. But then I don't know, what's happening is that ethical conduct. There is an ethical conduct. Very often, I mean, it sounds frivolous to very many scientists because it doesn't fulfill the category of utilitarianism. So it has to be useful. So who's measuring the usefulness? Who's measuring the utility? And is it enough that it's useful to human organisms, to human animals, right? Or how about ethical conduct with plant? How about ethical conduct? To which degree is the argument that it's making progress for human science to human benefit is an argument for a mouse to die? No? And there are many artworks, many of female artists who question that. There's Kathy Hayes' project where she did performances with lab mice that are programmed as knockout mice that are programmed to be killed, to die, to study the pathology that the artist herself was suffering from. So she rescued some of the lab rats and cured them so as a performance, to say, these are programmed to die to cure me, I cure them, right? Or Beatrice da Costa's piece, no? Dying for the others, is a piece where she's herself dying of cancer actually, but in the very phase of dying, and unfortunately Beatrice passed away 3 years ago, and in the very process of dying herself of cancer, she creating an empathy with the lab organisms designed to die to heal us. No? And this has a lot to do again with this post-anthropocentric view. No? Also, it's interesting to read these artworks as epistemic indicators. Let's take this work by Kathy Hay. Yeah? Healing the rats, curing the rats. But her last project is an epistemic indicator. So this was about human and animal relationship. We have many other projects of art with plant-human relationships. And you have more and more projects right now, and this is my paper next week in Montreal, about relations that we have to microorganisms. And the new project by Kathy Hay is actually to become David Bowie. It's called Becoming David Bowie. It's not that she's trying to sing like David Bowie, to perform or has the same haircut, but she's actually trying to get a microbiome transplant from David Bowie. So actually to have his gut bacteria and to make her gut like his gut. Because after, post-cognitivism, no? So in how far is our actually this human, after the Human Genome Project, you have the Human Microbiome Research Project now. So because genetics was indicating us that maybe there are so many organisms in our bodies that we do not know, because you cannot culture them outside of the body. But the genome has told us that there must be some, because you have a real genome, and it's not your genome, it's coming from something else that is in your body, no? So studying that and seeing a lot of bacteria must be there, but you didn't see them before. So, and this is where the research is going now. And this is interesting, from the code-based genetic approach of something that is encoded to a more messy one of how do these bacteria actually work together. because they outnumber human cells in our body by 10. Yeah? There are much more bacterial cells than human cells in our bodies. So that's why I said that a lot of these artworks can be read as epistemic indicators, no? And they point to directions that pick them up very early, and they have a quality as such as well.
I have one practical and maybe provocative question. This kind of big, huge expositions are really expensive. How and where to find money to make such a great exhibition? Mm-hmm. Who pays this?
Yeah.
Yeah. Okay.
That's a big problem. Yeah.
Because I can imagine that such an exhibition like was synthetic, that it would be in Brno, for example.
Well, it wasn't that expensive compared to what normal museum budgets are. The white cubes are quite rich, huh? Still. The white cube galleries, but they are not really interested. Oh, it depends. For example, in Luxembourg, when sculptor faces went to Luxembourg, it was in fact a place, a typical normal art, contemporary art gallery, but they were really interested in this. And I think the Luxembourg budget was a normal museum budget, let's say 250,000 euros for 25 artists. That's a middle European museum budget for sure. But we can do for something for nothing, right? It depends on how good your infrastructure is. So if you don't have a lot of money, you have to invest more time into building infrastructures, getting people to help you, convincing scientists and companies to bring you the stuff, and then you can make something for 10,000 euros. And this synthetic exhibition wasn't that expensive. No. Also, I was painting the walls myself, I was laying the cables, and then that makes it much cheaper, right?
Yeah, something once I was writing about a couple of papers, I think.
Yeah.
About the way how art exists, how art survives this.
I mean, I prefer to have a gallery technician who's doing that, but it depends, and if you don't have it and you want to make the show, you end up painting the walls all night, and so it's okay.
Yeah, because I think it's probably also like new media art in general, that you don't have, it's not part of that creative industry project of European Union, because you don't, it's probably not art for the bourgeois or the middle class who will go for the dinner and who will go to buy new dress, and I don't know, who will pay for a bottle in some city to go there. So maybe it's like small group of interested people, or maybe you can tell us something about the audience who will come to see these exhibitions, like scientists or
it depends where it is.
Yeah.
I mean, my attitude is to make it, let's say, more arty in a more scientific context, and really more epistemologically in art spaces. And sometimes artists do not necessarily agree with that. That sometimes artists want to be in a certain system, and many artists do not want to be branded at all. I mean, I hate this label bioart. It's completely anachronistic to me, and it has been abused by many just to hop on the opportunity of a bus that is departing, and you want to hop on the bus, so we call it in a certain way. But as a label, for example, it would prevent you from going to a white cube system, right? So many artists, I also even know artists that do not submit to Ars Electronica anymore because it's still systematically split, right? It's going there, you know? That artists on both sides are more and more merging. So when you go to a media art festival, lots of pieces can be nice gallery pieces now. They're not just body collage. No? And the Bois pieces are actually in white cubes, right? The shitting machine.
And I have to say that for me it was very interesting the way how you developed the concept of the representation or staging of the aliveness. I think it worked very well. That's why I thought that you had the art history education, because I think it worked very well for the white cube. But on the other hand, it can be interesting also for people educated in biology. But as you said, maybe they feel like it's what they are doing there in the laboratory if they don't have any result or they don't seek for any new medicine to heal human or whatever. So maybe the audience would be still the audience of these white cubes or the art from the art.
Well, but also scientists come to white cubes, right?
Yeah, they like very much classic, but they like the classic paintings usually.
But this is changing, no? And for example, these two residencies I was talking about in the very beginning, they are actually, one of them was done with a project from the European Union. No? This is an energy project where the art has one of 15 other building blocks. And according to Markus Schmidt, who is the PI, the principal investigator in this project, the art part was the easiest to get through. All the others had to rewrite something. The art part was just fine because people really wanted to do this. So, and the scientists in Denmark really wanted to be challenged by an artist to say, I want to learn something. And the artist is not just somebody who's going to visualize my studios or no, to have a challenge and a real discussion. To think about something new. No? So. And likewise in Los Angeles, the scientists very freely proposed that, yeah, I'm interested in this, let's say, I would like to work this with artists. It's so incredible what this Thomas Feuerstein, for example, does.
It's true that I remember I was on one conference of artificial intelligence, whatever, and Liebeck was presenting there his work. And scientists were laughing all the time, and I didn't understand to it because it was too complicated for me. But there were jokes on their kind of dogmas of the discipline, I think. So I think it's really important somehow to find also the way how to present it to the audience, that they should know more about what's going on in the laboratories with the bioengineering
well, Louis Beck knows a lot about this, by the way.
Yeah.
But Louis Beck has a kind of holistic approach to it through biosemiotics. And usually scientists laugh at biosemiotics because holism is not in fashion, right? But very often what happens you talk to a specific scientist on a specific problem, and you make a link to something what you know about in a side area or a kind of neighbor field. And the answer is very often, oh, I don't know anything about this. You know, I'm just here, right? And I know a lot about this. So, and when scientists sometimes laugh at something, I mean, there is also a questioning, how can you link this to this? No? And yeah, it's a risk, but we also live in a culture of over-specialization. And sometimes such art projects can help us thinking of the whole, no?
Out of the box, yeah. Some more questions. So I think if not, thank you very much once more for your interesting lecture and the time you spent with questions.
Good, and hopefully we have a lot of beer or wine and can
yeah, well, thank you.
Thank you.